Okay, so first thing it asks is just to define the difference between organic versus inorganic. Remember, when they talk about organic things, we're thinking of living matter, living material, and it's usually carbon based. And then inorganic would be things that are non living, like rocks, water, elements that don't contain carbon that are not part of macromolecules or living organisms. So, again, think living versus non living, carbon based versus not. Like salt would be inorganic, and an amino acid would be organic. All right, what properties of carbon make it central to life on Earth? Important things about carbon are that it has the ability to make four bonds because it has four valence electrons. It can make double bonds, triple bonds, single bonds, obviously. It can make ring structures. It can make really large molecules. Um, and that's pretty much one of the reasons why it's central to life on Earth. Four classes of organic macromolecules are carbohydrates, lipids, proteins, and nucleic acids. And so we're going to go through those. Remember, carbon, another quality about it is that it can host a variety of functional groups. The most important functional groups you need to be concerned with are ones that contain oxygen. So we talked about the carbonyl, the hydroxyl, the carboxyl. All of those are going to contain oxygen, and that oxygen is going to make those molecules polar. So remember, the more oxygen surrounding a molecule, the more polar it is, and the more going to be able to interact with water. So we have a saying like dissolves like. So if it's a polar molecule, it would be dissolved in water, which is also a polar molecule. And remember, that's because oxygen is highly electronegative, meaning that it's going to be pulling the electrons. And so when it pulls the electrons, it has a partially negative charge, making it kind of like charged. And that way, water is going to be able to interact with it and actually help dissolve it in solution. So anything that contains oxygen is most likely going to be able to dissolve in water. And the more oxygens it has surrounding the molecule, the more water is going to be able to interact with it, the more it's going to be able to be dissolved. So if we ever look at a sugar, let's see if I can write. Nope. Okay. So if we ever look at like a sugar molecule, it's going to have a lot of OH groups on it. It's surrounding it. And that means it's going to be able to dissolve in water because all those OH groups are going to interact with the water and allow it to be dissolved, which is why sugar dissolves in water. And so any group containing oxygen is going to help the molecule dissolve in water. It'll be polar. It'll be hydrophilic. Any group that is mostly carbon-based or like a long carbon chain, a ring, anything like that is going to cause it to be nonpolar. It would be hydrophobic and it would not dissolve in water. And then let's see, sulfhydryl groups, those contain sulfur. Those are going to be important in the amino acid cysteine because you're going to be able to create those disulfide bridges. Remember, the carboxyl group and the amino group are both found in an amino acid. The carboxyl group is an acidic group, and the amino group is a basic group. And so the amino is the amino group, and the acid is the carboxylic acid. And both of those make up the backbone of an amino acid. So those are kind of some of the things that you want to remember about functional groups. And so you have a bunch of notes in those. Maybe you can go back in your binder and look at those again. But just understanding that if they have a charge, they're going to dissolve in water. If they have no charge, they're probably going to be hydrophobic. If they have oxygen, they're going to be polar and dissolve in water. Things like that are important to remember. Monomer versus polymer. Remember, polymers are large organic molecules that are made of repeating units called monomers. So if you think of something made out of Legos, the repeating unit would be the Lego, and whatever you create out of the Lego, out of the individual Legos, is your polymer. So let's say you made a Lego building, then each individual Lego is the monomer, and the polymer is the building. So we have, let's see. If you guys remember, dehydration synthesis is when we have two molecules next to each other that we remove a water molecule, and that allows them to join. So we would have something like an amino acid. So I think I can write H2N. Then we have our central carbon, and then we have our carboxylic acid. And then we have our R group. that is going to be next to another group that contains just a hydrogen. And I'm just going to write a squiggle. And remember to join these two molecules together. This just is going to run off my screen. We would remove a water molecule. And so we would remove H2O. So we have two H's and an O. And so they would be oriented like that. And so we would take away this H2O and we would join the molecules together. So this is what we did when we made those macromolecules that I hung around the room. One group cut off the OH and the other one cut off the H, and then we put them together, and then we glued the H and the OH on the water droplet to represent that water had been removed. Okay, so that's dehydration synthesis, which is important for forming molecules. So to form a molecule, you remove water, and to break a molecule, you add water. So hydrolysis, hydro is water, and lysis is like to split. So hydrolysis is using water to split. And so that's how we would break down these large organic molecules. So after whenever it's time to make large organic molecules, we would use dehydration synthesis. And when it's time to break them down, we would use hydrolysis. So when you eat food, you break them down using hydrolysis, and then whatever your body needs, it's going to reassemble those monomers into polymers that your body needs using dehydration synthesis. So let's say you eat a cow, you get protein from it, you can break down that protein into amino acids and then reassemble those amino acids into whatever your body needs through dehydration synthesis. So that's why we drink water, and that's why we excrete water. Okay. Highlight the components of the mnemonic that relates to chomps. CHONOPS. I've seen it CHONPS. I don't know why I have it like that. But remember, you need to know kind of the basic elements that are found in each. Remembering that nitrogen is found in like amino acids is important. Phosphorus is found in DNA. Sulfur is found in proteins. And so sometimes they'll use this information to ask questions about, at the end of all of this, they purified material and they saw at the end of an experiment, they purified the material and found sulfur in it. Is it a nucleic acid or a protein? Well, you would say, well, DNA doesn't contain sulfur. And so it would have to be a protein. Or they found phosphorus in it. Is it a nucleic acid or a protein? And you'd be like, well, it'd have to be phosphorus because DNA is the only one that contains phosphorus. And then again, same with nitrogen. Nitrogen could be found in nucleic acids and it can be found in amino acids. But it's just good to know like that carbohydrates and lipids are only made of carbon and hydrogen and oxygen. But the other ones, proteins and nucleic acids, have different elements that distinguish them. Monosaccharides, I'm just going to go over one. Glucose is a good monosaccharide to know. It's a basic sugar. Remember, glucose and fructose together make sucrose, which is common table sugar. And we know that glucose is super important for things like cellular respiration. We break it down through a series of steps to create ATP in your body. The bonds, the course exam description did want you to know, like, amino acids come together in a peptide bond, and carbohydrates have, what's it called? Can't remember. But all the names of the different bonds off the top of your head. But we probably aren't going to need to know that for just the FRQ type test we're taking. So you might want to look over that. Like I said, I can't remember everything. I know that nucleic acids have a phosphodiester bond, and for some reason I just can't remember the name of the carbohydrate bond right now. But I don't think that's going to be a huge thing. Isomers means they're all made of the same elements. So these all have the same molecular formula, C6H12O6, meaning they all have 6 carbons, 12 hydrogens, and 6 oxygens, but they're arranged differently. So they, again, if you're using a Lego example, everyone used 12 Legos, but this person used 12 Legos to make a ship, this person used 12 Legos to make a building, and this person used 12 Legos to make a dog. I don't know. But basically, you use all the same materials, C6H12O6, but they're arranged differently. And we know that if you have a different structure, you have a different function. And so that's what an isomer is. Um, Carbohydrates, some of the main polysaccharides that we talked about that you might want to look up. Again, those one-page reference doodle diagrams that I gave you have a lot of information. I'm just going to go over 4 that come to mind. You have starch, which is storage of energy in plants. You have glycogen, which is energy storage in animals. You have cellulose, which is structural support in plants. And you have chitin, which is structural support in animals, specifically arthropods. And remember, all of those are made of repeating glucose monomers, but the way that the glucose molecules are arranged determines whether they are um, energy storage or are they structural support. Remember, we can digest the ones that are energy storage, but we cannot digest things that are structural support. So even though they're both made of glucose, depending on the way they're arranged and the way the glucose molecules are linked determines whether or not the enzymes in our body can actually digest them. And again, that's because structure equals function. And so if the structure is different, the function is going to be different. And if the structure is different, that means that our enzymes might not be compatible with those structures when they go to break them down. And so that's just kind of what I was talking about. Different structure, different function. Our enzymes in our body can digest starch and amylose, but we cannot digest fiber and cellulose because they have a different shape. So they have a different way of interacting with our enzymes that have a specific active site. Lipids are made of carbon, hydrogen, oxygen. They're not really considered true polymers because you can have all different types of lipids. You can have triglycerides, which are fats. You can have steroids and hormone molecules, and they all kind of look a little different. So they're not a true polymer, but because they're not just like one repeating unit. You can have ring structures and all these different things that make them different. Um, okay, so like I said, we have for the different types of lipids that you could think of, we have steroids, we have fats, we have phospholipids, which make up the structure of our cell membrane. We have waxes. Those are the big 4 different types of lipids. And so the ones that are the most relevant to us are going to be triglycerides, knowing that we have the saturated triglycerides, which are solid at room temperature, and unsaturated triglycerides, which are liquid at room temperature. And then we have phospholipids that make up the membrane, where they have the hydrophilic head and the hydrophobic tails. And that's going to cause them to create that bilayer, which creates a membrane for us. Also, remember that the fact that the saturated fats are more solid are going to influence the membrane as well. Because if the phospholipid tails are saturated, that means it's going to be more solid and the membrane is going to be less permeable. As opposed to if the phospholipid tails are unsaturated, that's going to cause the membrane to be more liquid. And when it's more liquid, it's going to be more permeable. So they could ask questions about the permeability of the membrane based on how saturated or unsaturated the tails of the phospholipids are. Also, remember cholesterol is a lipid and it's embedded in the membrane, and it's going to help create a buffer to make sure the membrane doesn't get too liquid or too solid. So those are different factors that can influence the permeability of the membrane. So those are important. Oh, so here's the difference between saturated and unsaturated. I got a little ahead of myself, but saturated have single bonds in the between the carbons and hydrogens in the long chain, and they're solid at room temperature. Unsaturated contain double bonds. They're liquid at room temperature, and again, those are going to cause the membrane to be more solid or more liquid depending on the content. Saturated fatty acids are not as healthy for us because just think of it as like they're solid things that are going through your arteries. It's less healthy for you. So things like butter, lard, they're considered saturated fats. And then unsaturated fats are things like oils, olive oil. Avocados have unsaturated fats because they have like type of oil in them. And so those are considered healthier for us. And I always just think about which one would you rather have running through your veins, solid stuff or liquid stuff? Proteins. Remember, proteins are made of amino acids, which have an amino group and a carboxyl group, which is why. The amino group contains nitrogen. So proteins have carbon, hydrogen, oxygen, nitrogen, and they can have sulfur. You can look up a diagram of an amino acid. It's got the carboxyl and amino group. The directionality in the protein is just the carbon, the amino and the carboxyl group need to, create like a line. The R group represents a variable group that can be attached. There are 20, sometimes people say up to 22, but there's about 20 different R groups, which makes 20 different amino acids. So that's just, they have a constant backbone with the amino group and the carboxyl group, and then the variable group is an R group. So if you think of it in algebra, it would be like X. So just like a random variable. And in this case, there are 20 different possibilities that R group can be, which make up the 20 different amino acids. So if it's just a single hydrogen, the molecule is glycine. And if it has more complex structure, then it can be any of the other different amino acids as well. The name of the bond between two different amino acids is called a peptide bond. And we can look at the R group to determine if the amino acid is polar or nonpolar. And remember, that's going to determine the folding of the protein. Okay, so remember that the R group is going to determine whether the amino acid is polar or not. And then when the when the protein goes to fold, then the R groups can be attracted to each other and cause it to have this really specific folding pattern. And so for each of the amino acids, indicate if it's polar or nonpolar, hydrophilic, hydrophobic. So here is our amino acid backbone. We have our amino group, which I'll circle here, and we have our carboxyl group here, and this creates a backbone that is repeating in all the different amino acids. Then you have your R group, which I'm going to put in red down here. All of these are the different R groups. And so we can look at the R groups and see if they have different elements in it to determine if it's polar, nonpolar, that kind of stuff. So I look right here and I see this contains an oxygen and a nitrogen. So both of those are highly electronegative elements, and they're going to cause this group to have a partial charge. So I would say that this one, I'm going to make them blue, which I'm going to P for polar. And polar means it's hydrophilic, it's going to dissolve in water. This one is going to be nonpolar or hydrophobic because it only has carbon in the R group. This one, second. Sorry, I keep having to go put Ansley's passy in her mouth. Okay, so these two, I really wouldn't classify them as hydrophilic or hydrophobic as much as I would just look at that sulfur and say, okay, these two have sulfur residue. They're going to be able to create kind of a bond, which we call a disulfide bridge in between them, which is going to create a cross link when the protein folds and helps stabilize it. So the more disulfide bonds you have, the more bridges that you have, because the more sulfur you have, the stronger the fold of the protein is going to be, because it's going to hold it together tighter. So it'll stabilize our protein structure because it has more bridges holding it together. So if you guys remember, we did our protein folding with the pipe cleaners and the beads. We put all the amino acids on the pipe cleaner. And then we started talking about, okay, well now all of the amino acids have their own individual properties. So the pink ones are negatively charged and the purple ones are positively charged. So all the pink and purple ones would bond. And then we put a paper clip in between them to represent the bond. And then this one, all the yellow ones have sulfur in them, and the sulfur ones are going to create a disulfide bridge. And so you would take the yellow ones and you put a paper clip in between those, showing a bridge that had been created. And the more paper clips you have, the more bonds you have, the more stabilized your final structure would be. Hold on. Okay, let's look. The sulfhedral bridges can form in between the sulfur residues. And then where would the dehydration synthesis bond forms? They would form between the hydroxyl group of one and the hydrogen of the amino acid next to it. So if I were to show that, I'm just going to continue using blue because remember, dehydration synthesis forms water. So we would remove one hydrogen here and one hydroxide there to create H2O. Hold on. Okay, let's keep going. So those are some important things to understand about the R groups of proteins. As we continue, the 4 levels of protein structure. Remember, those are going to be important to understand how proteins fold because it's going to determine their shape, which determines their function. So, you just want to see everybody? You want to say hi? She's swaddled in her little burrito. Oh, hello. Oh, you look crazy. Look at how I'm bald at the top. Like a little old man. I'm a month old tomorrow. Stop crying so I can finish this video. Okay. She thinks she wants to eat, but she's not due to eat for another hour. No bottle yet. Okay, 4 levels of protein structure. So again, I always think about this as the activity we did with the pipe cleaners with the beads on the string. So the first level of protein structure is just the sequence of amino acids, so the sequence of beads that we put on the string, and that's coded for by the messenger RNA. Remember, that's translated at ribosome, and then the ribosome is going to read it, and it's going to create the sequence of amino acids. So that just represents the beads that we put on the pipe cleaner. Secondary structure is when you start to have the alpha coils and the beta pleated sheets, and that has to do with just interactions between the carboxyl group of one amino acid and the amino group of another one. So remember that the alpha group is going to have this coily structure, and beta is going to have like a pleated sheet. And so again, that just happens between the carboxyl group of one and the amino group of another. In the backbone are just going to be attracted to each other, and they're going to cause this repeating alpha coil or beta pleated sheet. Tertiary is when the R groups actually start interacting with each other. Sorry guys, hold on. Okay, let's keep going. So the secondary structure has the alpha helix and beta pleated sheet, and that's just the interactions of the backbone. And then that would just be when we took it and coiled it, and then we folded it so that it had those coily shapes. And then lastly, we have, or second to last, we have the tertiary structure, which is going to be the interactions between the R groups. So that's what, I'm sorry, hold on. Okay, sorry. Clearly she finds my lecture on protein structure very boring. But let's continue. So then we get to tertiary structure, and that's the interaction between the R groups. So when I talked about putting the beads on the string, the different colors would represent the different amino acids that have different properties. So a polar group might be attracted to water, and so it would fold towards the outside, and a hydrophobic group would go towards the inside. And one with a sulfur group would find another one with a sulfur group and create a disulfide bridge. And so that's going to start giving that 3D complex structure. And we represented that by using paper clips to bond them and giving it like a 3D structure. And then lastly, we have quaternary structure, which is just when one protein interacts, or one polypeptide that's folded interacts with another polypeptide that's folded to create a functional protein. And so if at tertiary structure, when it's all folded, it's a functional protein, then it's good to go. But if it needs to interact with another polypeptide chain to become a functional protein, then it would need to have its quaternary structure. So not all proteins have a quaternary structure, but if they're not done at tertiary and they need to interact with another polypeptide chain, then they would have a quaternary structure. Chaperonins are molecules that help guide the folding of the proteins. So if we were doing that protein folding activity, then I guess your hands would represent the chaperonins that were helping guide the different R groups to interact with each other. Denaturation of a protein can be caused by changes in pH. It can be caused by changes in temperature. Remember, extreme heat would melt the bonds that hold the protein together in its complex shape. A change in pH is going to cause the protein to also be denatured because some of the R groups are acidic and basic, and if you change the pH, it's going to change the way that the basic and acidic R groups interact with their environment and cause it to change the way it folds. Nucleic acids, remember, they have carbon, hydrogen, oxygen, they have phosphorus. and they don't have sulfur. And they do have nitrogen as well. A nucleotide, remember that it is going to be a 5-carbon sugar. It can be ribose in RNA or deoxyribose in DNA. It has a phosphate, and then it has the base. And remember, the base can be adenine, thymine, cytosine, or guanine in DNA, or adenine, uracil, cytosine, and guanine in RNA. They have a phosphodiester bond. And remember, they run 5' to 3', so the 5' carbon is going to be at the top, and then the 3' carbon is going to be at the bottom. So looking here, if we were doing, this is a bad diagram of DNA. Hold. Okay, sorry. So this is a bad diagram of DNA, especially if you're labeling the 5' and 3' ends. So when you look here, one side should have a pentagon that's like that's kind of like a normal pentagon, right side up. And we have 1, 2, 3, 4, 5' carbon at the top. And then these pentagons over here should be upside down. So this one should start with phosphate. So I pulled up a better DNA diagram. You can see here, here we have the pentagons that are the correct way. This is the 5' to the 3', and then the ones that are upside down will be running 3' to 5'. Remember, that's called anti-parallel, where the strands run in opposite directions. That's important when we get to things like DNA replication, which determines the leading and the lagging strand. So, sorry, let's get back. The differences between RNA and DNA is that RNA contains uracil, it's single-stranded, and that DNA is double-stranded. It has equal amounts of adenine, thymine, cytosine, and guanine, and then it has thymine instead of uracil. So yeah, and remember, sorry, I just saw that it wanted us to show where the hydrogen bonds occur. That's going to be 2 between adenine and thymine and 3 between cytosine and guanine. Remember, it's easier to break the bonds between adenine and thymine because there's only 2 hydrogen bonds as opposed to cytosine and guanine, which have 3 hydrogen bonds. So the directionality of this is going to go 5' to 3', and then this should actually be upside down, being 3' to 5', and that is anti-parallel. So that is all of the macromolecules information that you need to remember. Sorry, Ansley was determined to derail my lesson, but that's going to be kind of the basis of what you need to know. And those are probably just going to be foundational information for setting you up for things like, DNA replication, understanding that kind of stuff. Protein folding gets into like enzymes and everything else.